Hello and welcome to Stars, Cells, and God. My name is Hugh Ross, and I'm joined by Fuz Rana, the president of Reasons to Believe, a biochemist. My background's astrophysics. I'm the former president. Fuz, you got a really good discovery on what's happening in the early Earth and what's going on with nitrogen chemistry.
Yeah.
I'm going to be talking about the most metal-poor stars astronomers have discovered and how that gives evidence for a cosmic creation event. And by the way, welcome to joining all of us here. We always want to welcome you into the discussion here. But Fuz, why don't you take it away with your discovery?
Yeah, well, Hugh, as I was thinking about this discovery, it caused me to reflect a little bit about how astronomy is actually unique among the historical sciences. I think you would agree that astronomy is a historical science.
Well, it's the only direct historical science because we astronomers are completely ignorant of the present.
Yeah.
I keep telling my wife that. I can't be accountable for what happens in the present because all of my data comes from the past. It takes like time to travel from stars and galaxies. And so, but the one beautiful thing about that, we have direct access to the past events in the history of the universe, which is why you get such compelling evidence in astronomy about the cosmic creation event. We can actually look back and see it happening.
Yeah. And that was the point that I was going to make is I couldn't have made it better is astronomers are literally observing what's happening in the past. Whereas every other historical science, there's data, there are clues as to what's happened in the past, but you can't directly observe it. So you have to reconstruct scenarios that are consistent with the data that you have. And that's where this particular discovery comes into play, because one of the big questions in origin of life research is when did life appear on Earth, and what were the very first life forms like on Earth? And there are some remarkable evidence from the oldest rock formations on Earth that clearly life was present 3.8 billion years ago. This is from a number of geochemical signatures. Also, there are microfossils. There are macroscopic fossils, stromatolites, fossils of microbial mats. There might even be indications that life originated even earlier than 3.8 billion years.
Yeah, there's some isolated places on the Earth, I think in northern Canada, with the same, maybe as far back as 4.0 billion years. But it is disputed evidence.
Yes.
By the time you get to 3.8, it's not disputed anymore.
That's right.
And moreover, that's when you got life continuously existing on the Earth, rather than, say, sporadic appearances.
Right. And so from that geochemical evidence, there's a very interesting picture that starts to emerge that I think is problematic for chemical evolutionary scenarios, but fits with the notion that there's a creator, and that is life seems to appear very early without any kind of real time for chemical evolution to transpire. The very first life forms appear to be single-celled, but they are biochemically and metabolically quite complex. And there appears to be a diversity of life on Earth, where there's a number of different metabolic lifestyles that seem to be present on the Earth.
So not just one species of bacteria.
Yes, exactly. Yeah. And that's the type of signature that you would expect for a creator. But there's still, a lot of questions as to what was the metabolism like. There are geochemical signatures that are characteristic of oxygenic photosynthesis, anoxygenic photosynthesis, sulfate reduction, other types of chemoautotrophic pathways. But again, the evidence is highly suggestive, but it's not conclusive, open and shut. And along those lines, one of those pieces of evidence is nitrogen-15 enrichment in rocks that date as far back as 3.2 billion years ago. And these are in a range that you would expect for nitrogenase activity, which is an enzyme that converts nitrogen in the atmosphere into ammonia, which is a critical process for making nitrogen bioavailable. That signature suggests nitrogenase activity was present. It's in association with microbial mats, which further gives support for it. Other more recent signatures like that in rocks that are 2 billion, 2.5 billion years in age are taken as evidence for nitrogenase activity. But it's still, again, there's still some uncertainty.
Well, let me clarify, because you said that it was enriched in nitrogen-15.
Right. Just it's a bit complicated. It's because the organisms actually favor nitrogen-14 when they fix the nitrogen.
Right.
And what's left behind is a residue that's slightly enriched in nitrogen-15. So it's a little bit different than carbon fixation, where it's the lighter isotope. In this case, it's the heavier isotope that shows the enrichment.
Well, thank you for that clarification, because I thought this is going to throw people. Doesn't life favor nitrogen-14? But this is the residue.
Yeah.
And so you expect the residue to be favoring nitrogen-15.
Right.
That's the evidence that is biologically derived.
Right. And so, it would be provocative if you had nitrogenase enzymes that would be present that early on. And I've got a slide showing a depiction of the nitrogenase enzyme. It's a very complex enzyme consisting of multiple subunits. And in fact, it's actually two enzymes that are complex together. They are working in tandem to take nitrogen and convert it into ammonia. Now, this is not, from a chemical standpoint, a trivial process. And in fact, this enzyme has two metal centers, an iron-sulfur cluster, and then there is a iron-molybdenum cluster that are, again, working in tandem in a stepwise manner to convert nitrogen into ammonia. And just to give you a sense for how remarkable this enzyme is, there is a process, it's called the Born-Bosch process, I believe, where you convert nitrogen into ammonia in a chemical setting.
So you're telling me it takes a molecule this complicated to make it work where life can use it?
Yes, exactly. Yeah. And this particular chemical process involves transforming nitrogen into ammonia, but it takes temperatures of about 400 to 500 C and about 150 to 250 atmospheres in order for that because the triple bond in nitrogen is so strong, to break it is really, really difficult. And what this enzyme does is very elegantly breaks the bond one at a time by adding high energy electrons to the bond.
And does this at a reasonable temperature?
Yeah, at room temperature.
Right.
So this is a remarkable enzyme. And, this enzyme occurs in bacteria primarily. So with plants today that are able to, some plants form a symbiotic relationship with a bacteria called rhizobium, which does the nitrogen fixation. There are other bacteria, these are called azotrophs, will also fix nitrogen, like Azobacter, for example. And cyanobacteria are the primary nitrogen fixers in the Earth's oceans, where they are, again, carrying out this reaction. The thing is, with cyanobacteria, they also are engaged in photosynthesis, so carbon fixation. The light reactions of photosynthesis generate oxygen. Oxygen destroys one of the metal centers, the iron-sulfur metal center in nitrogenase. So oxygen is a poison to nitrogenase, but cyanobacteria do both. And they have a very elaborate process where they regulate the production of the enzymes involved in the light reactions of photosynthesis. So at night, those reactions are shut down. That's when the nitrogenase enzymes are expressed and nitrogen fixation takes place. So there's a very elaborate control of coordinating photosynthesis with nitrogen fixation that's necessary. So it's not just simply this really complex protein, but it actually is a detailed coordination of the activity of another very complex metabolic process.
Any idea how the clock works?
I'm not completely sure.
I didn't mean to put you on
No. I'm sure people have studied this. I'm not completely sure how it works specifically. There's got to be obviously proteins that are light sensitive that are regulating gene expression and shutting down gene expression and activating gene expression at the right times in order to coordinate this, but precisely how it does that, I don't know off the top of my head.
Well, the fact that you don't know tells me that's another really complicated step in the process.
Yeah, or either that or I just maybe should be better read than I actually am.
No, I just think that's a really fascinating thing to look at. That clock can't be an easy thing to pull off.
So where this paper comes in, which was published in the January 22nd issue of Nature Communications by a research team from the University of Wisconsin, is, well, how can we confirm the signature for nitrogenase activity at 3.2 billion years ago? So there's a technique that researchers have been using for a while where they do ancestral reconstructions of proteins. And it's an approach that's really seeped within the evolutionary paradigm. And I've got a slide that shows kind of schematically how they go about doing it. And in effect, what they do for these ancestral protein reconstructions is they survey all the proteins that are from nature today, extant proteins today, and they build evolutionary trees, and then they try to infer what ancestral sequences for those proteins would look like. And a lot of times you have nodes on the evolutionary tree, so they'll build the reconstructed proteins on those nodes, and then they'll use that to go back further to try to get an idea what would these ancient proteins look like. Now, and then once they do that, they then produce a gene that corresponds to that protein. They incorporate it into a host cell that can express that protein, and then they study the biochemical properties of that reconstructed protein. So it's a very elegant process that people use. Again, it's steeped in the evolutionary framework, and you're assuming that the ancestral reconstructions are correct. It's the most probable amino acid sequence. So these could very well be proteins that never existed, but it's a way to get a window, into the past. So this research team applied this technique.
Now, how many proteins were they working with to start with?
Hundreds of them. They surveyed the entire library of nitrogenases, and they essentially, the next slide shows the evolutionary tree they built with all these different proteins.
Okay, now I can tell there's a lot of proteins.
Yeah, so they categorized them into group 1, group 2, and group 3. Now, group 3 have nitrogenases that instead of having molybdenum iron as the second reaction center, it has either vanadium, iron, and there's another one that has iron, and those are more unusual nitrogenases. The most common one is the molybdenum iron. So group 3 was these unusual nitrogenases. They threw that out. And group 1 was of interest because that's where cyanobacteria occur, which would be proxies for early life on Earth. So then they took group 1, if I could have the next slide, and they actually expanded that, doing a phylogenetic analysis, expanded that, and they noted that there were 4 nodes, and those nodes are shown there in the color, and those were what they used to do the ancestral reconstructions. Those represent the ancestral reconstructions. What would those proteins at these nodes most likely look like? And the next slide shows how they mapped these onto life's history. And so ancestral protein 4 goes back to about 2.2 billion years ago. So anyway, that's what they did in their analysis, and then they just analyzed the biochemical activity of these nitrogenases, and showed that the nitrogen 15 enrichment of all four of them was comparable to what we see for nitrogenases today, and would likely explain the geochemical signature in rocks that are 3.2 billion years ago. So they basically conclude that this is probably a bona fide signature for nitrogenase activity in rocks that are 3.2 billion years ago. Now, something else interesting too, and if I could have the final slide here, what they say at the conclusion of the paper is that this is a minimum age for nitrogenase activity. They say the similarity of the lowest nitrogen isotope values in early Archean rocks suggests that the currently accepted minimum age for nitrogenase activity at 3.2 billion years ago may be too conservative, and that its origin could predate what is currently resolvable in the rock record.
In other words, it's probably there in the very earliest rocks.
Yes, yeah. That's right. And given its critical importance for life, that makes sense. So, I mean, the obvious conclusion here is that, wow, this is even one more piece of evidence that life appeared very early in Earth's history, and it was complex metabolically and biochemically at the very outset. And that, again, is very uncomfortable in a chemical evolutionary scenario, and really makes sense in a creation model perspective, where we would predict, as part of our creation model for the origin of life, life appears early and it's biochemically complex.
Yeah, I'm impressed that wherever you have in the early Earth rocks and liquid water, you can see the evidence of life.
Yeah.
I mean, the thing about this Great Slave Lake in Canada, where it goes back 4.0 billion years, they do see a signature for liquid water and for rocks that are not molten. Yeah. And there is a strong suggestion chemically for life there.
Yeah.
So it's not stable. By the time you get to 3.8, it's stable. Yeah. But isn't it interesting? The moment you got rocks and liquid water, life is there.
Yep. Yeah, and, a lot of times, people that hold to a naturalistic, materialistic perspective would say, well, that just means life can originate really easily. But I think everything we've done in the lab suggests the opposite.
Well, just what you presented today, I mean, getting all this stuff to happen without any time passage.
Yeah.
So
now, putting on the hat of a skeptic, there's one more point that I want to talk about. Is a skeptic could come along and say, look, really compelling evidence that life appears early in Earth's history. It's metabolically complex. I think most original life researchers would readily acknowledge that point. But the whole evidence for the legitimacy of this geochemical signature for nitrogenase is predicated on an evolutionary analysis, right? That's what you're doing when you create these ancestral versions of nitrogenase. And so that's actually an interesting challenge, and is one that we need to engage. And my point would be, well, you could actually look at this analysis as not necessarily evolutionary in nature, but from a design perspective, where these ancestral proteins that you are presuming existed that represent the nodes could simply just be core designs that a nitrogenase in that particular group would actually possess. And so as you're going back in time, maybe what you're not really doing is generating ancestral proteins, but really you're winnowing down nitrogenase to its most core basic design, as opposed to an ancestral protein. And so in that case, still, I think the analysis holds, because you're saying, well, if you're looking at the nitrogenase chassis, this is still capable of producing that kind of signature.
Well, using the word chassis makes me think of automobiles.
Yeah.
I mean, today we got a very complex array of truck and car chassis, but you could look at the current assembly of all these different chassis and do the same thing and determine an ancestral history back to the original chassis. And I don't think you'd be far off.
Well, it's interesting that you bring that up, Hugh, because I did a little research in the literature to see if anybody has ever done a phylogenetic analysis on technology.
Yeah.
And it turns out that there are a number of papers that have been published by technologists who have done that very thing, where they've taken automobiles and they've done a phylogenetic analysis on them, and you can literally reconstruct the history of automotive technology in that way, right? But these are designed. These are not things that are the product of unguided
the original
process.
was designed.
Yeah, yeah. So that just simply reinforces the point that I'm making. And in fact, there are a couple of papers published that are used that the authors wrote them to be used in biology classes to teach students how to build phylogenetic trees using automobiles. Wow. So there's, again, an irony in that. But so the point is that there's actually a precedence where people are doing that very thing. So I think you could easily look at this ancestral protein reconstruction from a design framework and produce, again, maybe even a history of nitrogenases, but it's not a evolutionary history, it's a divine history.
Well, I'm wondering if this can be done. If you take these papers where they're doing, say, automobile chassis or automobile motors, I just think of how the motors have evolved over time.
Yeah.
I'm old enough to remember when double overhead cam engines were really rare.
Yeah.
Today they're everywhere, but that wasn't the case back in the 30s, 40s, and 50s. Yeah. So, taking these papers that you're citing about automobile phylogenetic trees and then using the analogy of what you're talking about here, it's like when you look at automobiles, you're looking at things that are, the phylogenetic tree is orders of magnitude simpler.
Yeah.
Than what you're looking at here. And no one in their right mind would say the phylogenetic tree of motors and chassis for automobiles is the product of naturalistic progression.
Yeah.
Clearly it's design.
Yeah.
How can you say it's not design when you're seeing something that's literally orders of magnitude more complicated?
Yeah. Yeah, I agree. So anyway, that's all I wanted to share with this discovery.
Maybe we can find a scholar or scholar community who can write a little article about these phylogenetic trees for automobiles.
Yeah.
I think that would be really fun.
Yeah, yeah.
So if you got those papers, let me know. I'll see if I can find someone to take that on.
I will.
That sounds great. Well, Fuz, that was remarkable and really makes a strong case. And makes me glad, too, that we titled our book Origins of Life rather than Origin of Life, because that's still an unsettled issue.
Well, that book was originally published in 2004, and we just did a reprint in 2014. And I've gone back to that book, and it's still remarkably up to date.
It is.
And that's, I think, not so much our prescience as writers as it is just the fact that the origin of life research is largely stagnant. The very questions that people were wrestling through 20 years ago now are the same questions that people are wrestling through today. Well,
I've noticed that at the 3 origin of life research conferences we've attended, Fuz, is like there wasn't a whole lot. 1999, 2002, was it 2015 or 2017? We went to one. And it's like every one seemed to be about the same.
Yeah.
So, and also knows the mood of those conferences was so different from scientific conferences I've attended, where people are saying, look at all the progress we've made. Yeah. They're all excited about the new stuff. Kind of what we saw was a mood of depression.
Yeah.
It's like, hey, we're in the same spot that we were 15 years ago, 18 years ago.
Yeah.
So, well, I'm going to switch gears here, Fuz, and going to talk about something literally orders of magnitude simpler than what you're talking about. So, and that's stars. The wonderful thing about astronomy, we get to observe things that are so simple, we can develop mathematical equations to predict their past and future behavior. You can't do that in biochemistry, but because our discipline, we're studying things that are so simple. Or as I often tell people, stars are just huge balls of gas, or almost all the gas is hydrogen and helium.
Yeah.
I mean, what could be physically and chemically simpler? So, but kind of a holy grail in astrophysics is trying to find the very first stars in the universe. And if you take even a freshman course in astronomy, you get introduced to what they call the 3 different populations of stars. Mm-hmm. Population 1 stars, population 2 stars, and population 3 stars. And we started off by saying astronomy is that one discipline where we directly observe the past, but it leads to some strange practices. So the population 1 stars are the ones that we kind of see now. Yeah. The population 3 stars are the ones that exist at the very beginning of the universe. So you think, why didn't they call the first ones 1 and the last ones 3? But it's the other way around. That's because the farther away we look, the farther back in time we see. Mm-hmm. So, but the core of Big Bang cosmology is that the universe begins with only one element. So you can imagine wearing a T-shirt where all you got is hydrogen on it. And that would tell you the original state of the universe. It begins with just hydrogen. But what happens during the first 3.5 minutes after the Big Bang creation event is that the universe is expanding. And as it expands, it gets cooler. I mean, it's a principle of thermodynamics. Mm-hmm. Any system that expands, the temperature goes down. And so the universe starts off infinitesimally small, but almost infinitely hot. But as it expands, the temperature drops, and there's a window about 20 seconds long where the universe passes through that temperature range where nuclear fusion can occur. Mm-hmm. Somewhere between 150 billion Kelvin and about 17 billion Kelvin. In that range is where hydrogen can be fused into helium. Think of the hydrogen bomb process. Yeah. So what happens during that 20 seconds, somewhere around 3 minutes after the cosmic creation event, is about 1/4 of the hydrogen in the universe gets converted into helium.
And there's also a little bit of trace lithium and a few other things, right?
You get lithium at an abundance level of 1/10 millionth of the helium.
Okay.
So we're talking a real trace amount.
Okay.
So yeah, once that 4 minutes is over, it's about 75% hydrogen, 25% helium, and then 1/10 millionth lithium. And some Big Bang creation models think there might be even a tinier amount of beryllium in there.
Okay.
Some even speculate boron, but most people think no, just the beryllium, and some say just the lithium.
Yeah.
But the point is, it's virtually 100% hydrogen and helium.
Yeah.
And then it's stars that convert that hydrogen to helium into carbon, oxygen, nitrogen, phosphorus, iron, the elements you need for life. So, but a core feature of the Big Bang model is the universe begins after 4 minutes with only hydrogen and helium and these extremely trace amounts of very light elements. You literally have no metals. So, and that's been a thing. Can we test that? And when you look at the cosmic microwave background radiation, it gives you strong evidence that indeed that is the case. Mm-hmm. But people have always been interested, can we directly observe stars that are what we call pristine before they get polluted with carbon, oxygen, nitrogen, iron, and calcium and other elements.
So you're going to have to look way back to the very earliest.
Way back in time. And what we've discovered with the James Webb Space Telescope is that, and physicists have known this for some time, if all you got is hydrogen and helium, it's really difficult to get small stars. And the problem there is the big stars burn up quickly. In fact, if all you got is hydrogen and helium, the stars are going to be 20 to a few thousand times the mass of our star, the sun. And say if it's 100 times the mass of our star, the sun, it burns up in 1 million years, which means none of these stars exist today. They're all burnt out. And when they're burnt out, you can't see them. So, and the question is, what about the small ones? Well, the small ones can't form unless there's a little bit of dust, carbon, nitrogen, and oxygen. But the thinking is, hey, these first stars that form out of pure hydrogen and helium, they burn up so fast, and because they're big, they end up as a supernova. And as a supernova, they explode the ashes from their nuclear burning into interstellar space. And so the thinking is, well, if you got small firstborn stars, they take so long to form, so you can get a small star forming that's virtually all hydrogen and helium, but it takes so long to form, some cases over 1 billion years, that it gets polluted by the ashes of the big firstborn stars. But in astronomy, we have this thing where we call a population 2 stars. These are stars that form after all the firstborn stars have formed. And so they're going to have significant enrichment from the hydrogen, or the carbon, oxygen, nitrogen, iron, etc., exploded by the firstborn stars. So they typically come in at, say, 1/10 as much material heavier than hydrogen, helium, up to nearly 1%. Those are the population 2 stars. And when they all burn out, you get the next generation, the population 1 stars. Our star, the sun, is in that category. And these stars are running between, say, 2 and 3% elements heavier than hydrogen and helium. So, but, you know, as a star is forming, it gets polluted by the stuff that's in the interstellar medium. Well, what astronomers have done is they determine, okay, if you got one of these small firstborn stars, as it's forming, it will gain a little bit of pollution from the remains of the really big stars that are firstborn, but it's going to be at a very low limit. In fact, it was a team of astronomers that calculated that it's impossible for a second-generation star to have anything less than 100,000 times 0.00001 times the iron content of the sun. So take the iron content of the sun, divide it by 100,000. No second-generation star can have less iron than that. They also get a limit for how much total elements heavier than helium you would have for a second-generation star. So this is how they identify the tiny stars that are firstborn, is that we know they have to have a metal abundance of elements heavier than hydrogen and helium below a certain level, and iron, which is relatively easy to see in a spectral study. We know the limit for that. And so astronomers have been on a quest to try to find these pristine stars that are still shining.
Okay.
And they have to be low mass, because after all, they have to be a star that's capable of burning for over 13.5 billion years. So our star, the sun, its burning time is 9 billion years.
Okay.
So it's got to be a lot smaller than our star, the sun. And if it's a lot smaller than our star, the sun, it takes a longer time to form and become a star, which means it's bound to have a little bit of pollution, but it's going to be a very, very low level of pollution.
Right.
Now, I'm old enough to where I realize we're talking 20 years ago, we didn't have the technology to take a spectrum of a star and detect elements heavier than hydrogen and helium at that extremely low level abundance. Today, we got the telescopes and the technology to do it. So kind of one of the quests of the past few years, literally this got started around 2020. That's the first time we had telescopes big enough and the spectral technology to actually pick these things up. And so we have now found 7 such stars. Well, Nature Astronomy a few days ago added to the list. We now have 8 of these stars.
Okay.
But what's interesting, and by the way, the only place you can find these stars is in the halo of our galaxy. I think the first slide I got here basically shows you our Milky Way galaxy, and then you see this very faint halo around it. And the point is, if one of these firstborn stars wanders in to where you have the stellar disk, it's going to get polluted to a degree that's going to be above that level. And so what astronomers are doing is looking for small mass stars in the outer halo of our Milky Way galaxy beyond the stellar disk, because there the density of stars is so low that we're going to be able to see this very low level of pollution and prove that indeed we're seeing a star that formed out of hydrogen and helium in the vicinity of much bigger stars that were forming out of hydrogen and helium. Which again explains why we need really big telescopes. Trying to see a really low mass star in the outer halo of our galaxy, there was a time when that was impossible. It's now possible for us to see stars there, small stars there. But again, it's extremely challenging even now to see stars that small, that far away in our galaxy. But the list now stands at 8. But this paper was published a few days ago. It's not still in the regular issue. It's kind of a paper published in advance of showing up in the actual journal, but it's been released to the public. And what's interesting about this star is that it has the chemical signature of being formed in the Large Magellanic Cloud, in the halo of the Large Magellanic Cloud, which is the nearest galaxy to our galaxy. It's it's a small galaxy. It's about 1/16 the size of our galaxy, but formed in the halo. And because the two galaxies are so close together, the star in the halo of the Large Magellanic Cloud wandered into the halo of the halo of our Milky Way galaxy. And if we could get the second slide up, maybe they're having a technical difficulty here. I actually show you a map here of our Milky Way galaxy, the Large Magellanic Cloud, and the Small Magellanic Cloud, basically showing you they're close enough together that our galaxy, being as massive as it is stealing stars and star clusters and gas clouds from the Large Magellanic Cloud and incorporating them into the Milky Way. Meanwhile, you got the Small Magellanic Cloud really close to the Large Magellanic Cloud. So for billions of years, the Large Magellanic Cloud has been stripping material away from the Small Magellanic Cloud. So the Large Magellanic Cloud is robbing the Small Magellanic Cloud. Our galaxy is robbing the Large Magellanic Cloud. But what's interesting to astronomers, the smaller the galaxy, your greater the probability that you're going to have surviving pristine stars from the very beginning of the universe, where the stars are formed out of hydrogen and helium. So this is the first time they've actually seen a star in the halo of our galaxy that they knew was born in the Large Magellanic Cloud. And what got the astronomers excited, they were able to measure the quantity of elements heavier than hydrogen and helium. It came in at 0.0000078 of the total mass of the star. And to put that in comparison, for our sun, it's 0.0196. So we're talking about a star that has got 28,000 less elements heavier than hydrogen and helium than our star, the sun. And there are stars that are clearly more metal-rich than our star, the sun. By the way, there's an interesting thing about our sun being at 0.0196. You get the greatest stability in the luminosity of a star when it's 0.020. And notice our star, the sun, is virtually right on top of that. In fact, it's within the error bar. So it gives us a reason why we happen to be orbiting a star with such extreme solar luminosity, stability. It's because we got the right quantity of elements heavier than hydrogen and helium.
So now that astronomers have an idea where to look for these population 2 stars, do you anticipate there being a flurry of discoveries? Because it seems like they now have a place to train their telescopes to seek them out.
Well, they've been training their telescopes since 2020 on the outer halo of our galaxy.
Oh, so this isn't new with this discovery then?
It's not new with this discovery. What's new with this discovery is that we now know this star was not born in our galaxy.
Okay.
It was born in a nearby galaxy, but that's a galaxy that's not as metal rich as our galaxy.
Okay.
So you're actually got the possibility of finding stars that are even less metal rich than the ones we see in our galaxy. And this wasn't in the paper, but what excites me is since Large Magellanic Cloud is stealing stars from the halo of the Small Magellanic Cloud, if we focus our telescopes, maybe we can find in the halo of our galaxy stars that formed in the outer halo of the Small Magellanic Cloud, which is going to get us really close to where the very first small mass stars formed. And that's going to actually not only tell us the Big Bang creation model is correct, it'll tell us which of the Big Bang creation models is correct. So it's another way to advance the discipline of establishing in more detail the Big Bang creation model.
Right.
And a lot of skeptics have said, hey, the Big Bang creation model, they haven't found these pristine stars. Well, now we found 7 of them.
Right.
And all of them are provably population 3 firstborn stars.
Right.
And we now have the possibility of increasing the database, and especially if we can get some from the Small Magellanic Cloud and actually see if we can establish the prediction. Are we going to see that the metallicity, elements heavier than hydrogen, helium, drops as we go from our galaxy to the Large Magellanic Cloud to the Small Magellanic Cloud? And it's going to be a bit challenging. I don't like a database of 1 because maybe that star wandered into the interior of the Large Magellanic Cloud. In fact, that caught my attention. We have found stars in the outer halo of our Milky Way galaxy that are more metal poor than this one that just got published a few days ago, which means it may have wandered into close to the interior of the Large Magellanic Cloud before it got sucked into our galaxy, which would explain why it's got slightly more metals than the most metal poor stars we've discovered here.
Yeah.
We're going to need a database of a few hundred to really determine this.
Yeah.
But bottom line is, this is another prediction of the Big Bang creation model that's been proven correct. And, years ago, a couple of years ago, we did a show here where we talked about how they found a gas cloud in the outer halo of one of these newborn galaxies that the James Webb Space Telescope detected. It was 100% hydrogen and helium. So it's like we've actually, and there was no stars in it. It was a gas cloud with no stars at all in the extreme early history of the universe that basically showed, the Big Bang got it right.
Yeah.
The universe begins with only hydrogen and helium. But these small mass stars with extremely low metals as an added piece of proof for that. And the bottom line is the Bible predicted Big Bang cosmology thousands of years before astronomers discovered that the universe has a beginning, that the laws of physics are immutable, where it's governed by a pervasive law of entropy or decay. Bible doesn't use the word entropy, but it does use the word decay, which I think is a reference to the second law of thermodynamics. And 11 places the Bible talks about the universe being a system that expands. And I've often been accused, Fuz, of saying, well, you're a 21st century astronomer. You're simply reading that into the text with hindsight. But there's an article up on our website where I document that Jewish and Christian theologians 1,500 years ago, 1,200 years ago, 1,000 years ago, 800 years ago, saw all these features in the Old Testament, including the idea that the universe is a continuously expanding system. And you can just simply go to our website. What does the Bible say about the Big Bang? We document the Bible verses. We also document what these ancient theologians saw in the text. So for them, it wasn't hindsight. They had no science to tell them that the universe had these characteristics. The Bible said it first.
You know, along those lines, I remember Paul Davies making a statement to the effect that he was deeply impressed with Augustine as a thinker because Augustine had this concept that time had a beginning.
Yes.
And that really is a concept that's part of modern day astrophysics, is the idea of time having a beginning. And of course, Augustine got that idea from the biblical authors, right?
He also spoke about space having a beginning.
Yeah.
So, I remember Paul Davies being stunned. Hey, the idea of a space-time beginning is something we had no scientific evidence for until the 20th century.
Right. And so, that reinforces the point that you're making. How would you respond to people that would say, well, we do live in a Big Bang universe, but just because it's a Big Bang universe, that means that the universe is expanding, but that doesn't necessarily mean that the universe had a beginning. That it's wrong to equate the Big Bang with the beginning of the universe. How would you respond to a skeptic that might raise that point?
Well, I had a couple of debates, Fuz, with two atheist cosmologists, and they basically have devoted their lives to studying this.
Right.
And they said, we've come up with all these models. In fact, they wrote a 400-page book, which I read, and we basically had a debate a couple of times about what they wrote in the book. And I think they were rather surprised. And I said, your book reads a lot like these Bible texts on what the universe has. And they said, well, aren't you aware, Hugh, that our models say the universe doesn't have a beginning? And I says, yes, but you redefine the universe and you redefine the beginning. Mm-hmm. It says, I see this with young earth creationists, where they cite Mark 10 and say, Jesus said, was it like this from the beginning? Yeah. And they assume that Jesus is talking about the beginning of the universe. He's talking about the beginning of human marriage.
Yes,
so it's the beginning of what? And, I remember listening to Fred Hoyle when I was a grad student, and he always capitalized the universe with a capital U, because his idea, the universe is the totality of everything.
Yeah.
And that's what I realized. These two cosmologists I was debating, they were basically defining the universe to be much more than the universe of matter, energy, space, and time we can detect. And they said, if we invent other dimensions of time, we can escape the beginning.
Yeah.
And I said, well, that's what Stephen Hawking did, and Hartle. They basically have postulated another dimension of time, which means you've got another time dimension in which you can have beginnings. But I said, that's not what the Bible talks about. Hebrews 11:3, the universe that we can detect did not come from that which we can detect. There is a beginning to what we can detect.
Yeah.
And they admitted in their book, and I cited what they saw in the book, what we call the, Arvin Borde, Alan Guth, Alexander Vilenkin theorem, the latest and most famous of the space-time theorems. They basically point out that even the inflation event has to have a beginning. Mm-hmm. And their comment was, well, if you go before the inflation event, maybe we can escape the beginning.
Yeah.
But now you're speculating beyond what we can detect.
Right.
And, in Christianity, God's beyond what we can detect, right? And so Hebrews is basically telling us the universe came from beyond what we can detect. So there is a provable beginning, as you see in the Borde, Guth, Vilenkin theorem. Yeah. There is a beginning to everything that we can possibly detect. Yeah. Not just what we can detect, but we can conceivably detect. Yeah. That's Hebrews 11:3. So you got to be careful in these discussions. What do you mean by beginning? Yeah. What do you mean by the universe? So what I was understanding was, hey, the way you define the universe, that would include the angelic realm with different physics. So I said, yeah, I understand. There are ways to get around the beginning if you're going to speculate, new constants of physics, new laws of physics, new space-time dimensions. But that's exactly what the Bible declares. Yeah. In fact, what I found ironic, what they were discussing in their book was virtually identical to what we were discussing in 2005 at Cambridge University. That was a time when there was 8 Christian physicists and astronomers and 8 Christians philosophers. And we were debating the beginning of the universe. And our debates were virtually identical what these atheists were having. So my point was they were on theological turf and didn't know it.
Yeah.
So,
well, thank you. Yeah.
well, thank you for bringing that up, because I notice a lot of people get struck when they say, hey, what do you do with these cosmologists that say they found a way around the beginning? But I said, yeah, look what they had to do to get around the beginning.
Yeah. 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 Well, thanks, Hugh. I appreciate that.
All right. Well, let's wrap things up here.
Sure.
Thank you for joining us today in Stars, Cells, and God. We post the video recording every Wednesday, so look for that. You can subscribe to our YouTube channel, the Reasons to Believe YouTube channel, and this is also available on other podcasts and media that are out there. And remember, the more we learn about science, the more reasons we have to believe that the Bible is the inspired and inerrant Word of God, and Jesus is Creator, Lord, and Savior. Thank you.